Okay, so I want to thank the organizer for the opportunity to speak and for all of you to staying until this time. And we're going to switch gears a little bit and have a tour inside the diabetic body. And so what is happening? So I have done some epidemiological studies in the U.S.-Mexico border, and I started studying tuberculosis, and very quickly I realized that it was impossible to study tuberculosis without understanding diabetes. So in my community, nearly 40% of the TB patients have diabetes, and as Carrie mentioned, about 1/4 of the TB cases, and about 1/4 of the TB cases are attributed to diabetes, in contrast to less than 6% attributed to HIV. So at a population level, diabetes has a much bigger impact on tuberculosis control than HIV. So with that, what I have been interested in doing is an extension of that and understanding why is a diabetic patient more susceptible to tuberculosis? And you know diabetes patients are more susceptible to other pulmonary infections as well. So what we learn from tuberculosis is going to teach us about diabetes in general. So how do I use the pointer?
Just the green button.
Yes, but it doesn't work. Okay, I'll use this one. So just to recap on the natural history of tuberculosis, when an individual is exposed to a tuberculosis patient, there are two possible outcomes. One possibility is that person never has evidence of TB infection. That's a major mystery. What is it about some people that have a very effective innate response that they don't, quote unquote, become infected? And then about half of the very close contacts become infected with the bacteria, but they're not sick. This is called a latent TB infection. About 1 in every 10 persons with a latent TB infection will have the risk of progressing to tuberculosis. So the question is, what is the impact of diabetes in this natural history of TB? So we know that it's about threefold overall. But is it a higher risk of a latent TB infection, or is it a higher risk of progressing to TB? How does that happen? And why is it important to understand the biological basis of the contribution of diabetes to tuberculosis? Well, first of all, it has clinical implications. It is very important to identify if a diabetic patient has latent TB. And the detection of latent tuberculosis is based on immune response. And as most of you may be aware, diabetes causes an immune compromise or immune dysfunction. So one of the questions that we had is, if we have a diabetes patient, will we be able to detect whether they have latent TB infection or not? Another big question is, you've heard from Anil and Carrie, the huge numbers of diabetes patients in TB endemic areas. And in TB endemic areas, we can have at least 50% of the individuals with this latent TB infection. So the question is, among the millions of diabetes patients with a latent TB infection, how do we stratify to determine in whom we should do preventive treatment for tuberculosis? So we obviously need to know more about the biological basis of this association between TB and diabetes. So we began our studies at the TB clinics with the TB patients, and our very basic hypothesis is that diabetes compromises the immune response to Mycobacterium tuberculosis. And specifically what we would expect is that a TB patient with diabetes would have a different type of immune response than a TB patient without diabetes when exposed to mycobacterial antigens. And then what we would expect is that the difference in this immune response will help explain to us what is it about the diabetes patient that makes them more susceptible to tuberculosis. So to do this, we did field studies in the TB clinics. We identified TB patients with and without diabetes. We drew blood. We incubated that whole blood with mycobacterial antigens for 24 hours. And if the patient, since these patients have been exposed to tuberculosis, within 24 hours, they're reacting and secreting cytokines. And so the next day, we collected the supernatants from this blood, the plasma, and we measured different types of cytokines. And then we compared, is there a difference between the diabetics and the non-diabetics to tuberculosis in the profile of cytokines that they secrete? Our expectation was that diabetes, you have an immune compromise, and therefore they will secrete lower levels of this type 1 cytokines. What we observed, however, was exactly the opposite. And this is an example. So this is the response to a mycobacterial antigen that is semi-crude, the PPD. And what we observed here, this is the glycated hemoglobin. And these are the levels, for example, of this cytokine, IL-2 as an example. What you can see is people who have diabetes that is well controlled or who do not have diabetes have a heterogeneous response with IL-2. However, it is very evident that as soon as people had hyperglycemia, chronic hyperglycemia, there was more interleukin-2 secreted. The same thing happened for interferon gamma. So we observed exactly the opposite of what we were expecting. And something interesting about that, we wondered, well, maybe we're using a semi-crude antigen. What if we use antigens that are very specific for Mycobacterium tuberculosis? Will we observe the same thing? And this has clinical implications because these antigens are the ones that are now being used in the new generation of interferon gamma release assays to detect latent TB infection. So we used the recombinant antigens from mycobacteria present in the quantiferon gold kit, the ESAT6 and CFP10. And here you see a table with the results. I have highlighted to simplify. So here are the two antigens, and we're comparing diabetes versus no diabetes. And there was significantly more secretion of interferon gamma in response to the recombinant antigens in diabetes or in people with chronic hyperglycemia. So the conclusion from this is that diabetes and chronic hyperglycemia, they do not compromise the response, the secretion of interferon gamma, or the response to one of these interferon gamma release assays. Not only that, but it enhances that response. This is a summary of a study done by other colleagues. It came out very recently. And what they found was essentially the same findings, that not only is there more T helper 1 cytokines produced, but there's also this other type of cytokines, T helper 17. So you can see here they were measuring the frequency of cells that were positive for interferon gamma plus IL-17 or IL-10 plus IL-17. And so, in summary, we're seeing something exactly what we were expecting. So we're seeing a hyperreactivity of the cell-mediated response to tuberculosis. What may this mean? So maybe this is a reflection of immune dysfunction. That is, we see more interferon gamma, more T helper 17, but we don't know if downstream from these cytokines, we are effectively killing the mycobacteria. These are not the ultimate killers of the mycobacteria. The other possibility is that in diabetes, there may be a higher mycobacterial load, which has been shown, and therefore there's more stimulation of the immune system. So what we're getting is higher cytokine levels because there's just more bacterial burden. So the problem with these studies is that we're studying patients here who already have developed tuberculosis. And so the good news is we're seeing differences in the response, but in order to understand what is it that makes them more susceptible, we need to go back to these early stages. So we have begun studies now focusing on this early stage of the response. And specifically, we want to study the impact of diabetes on the phagocyte responses to Mycobacterium tuberculosis. So we want to study the first encounter of Mycobacterium tuberculosis with the diabetic host. So of course, we cannot do this in vivo. So we are now doing this in vitro. So what we have done is that we collect blood from individuals with diabetes, not with evidence of tuberculosis, and we isolate their monocytes or phagocytes, and we isolate serum. And then in vitro, we add the mycobacterium, and we evaluate that interaction. And we are comparing the monocytes, of course, from diabetics and non-diabetics. So what happens is that the serum coats or opsonizes the mycobacteria and with either complement or natural antibodies, and that coating facilitates phagocytosis or the engulfment of the bacteria into these white blood cells via receptors for complement or receptors for the antibodies. Then, what's the fate of these intracellular bacteria? Mycobacterium tuberculosis is an intracellular bug. So it can either replicate, but an alternative is that it may be killed. So what happens in the diabetic monocyte? So we have begun, and most of what I'm going to show you is about our studies in this first encounter, which is the association or phagocytosis of the monocyte and the mycobacteria. And then I'm going to tell you very briefly about what happens in terms of once the mycobacteria is inside the cell, what happens inside the diabetic monocyte. And this is just an example of what we see in the lab. So when we incubate the monocytes with the mycobacteria, we wash away the mycobacteria that was not engulfed by the cells. We stain the nuclei of the cells blue, so each of these is a monocyte, and here are the mycobacteria. So we count the percent of monocytes that engulfed a mycobacteria. So that's our readout. This is a summary of what we observed in terms of that association. And so here are the participants, and they're classified by their diabetes status into those without diabetes and those with poor diabetes control, and in the middle, people with borderline diabetes. That means that they had either high HbA1c or hyperglycemia, but not both. And basically what we observed is that individuals who do not have diabetes, about 1/3 of their monocytes will have engulfed the bacteria. If we have a diabetes that is borderline, it's about the same percentage. We don't have any alteration in those monocytes. But if we have poor diabetes control, then the phagocytosis, that engulfment of the bacteria, seems really compromised in the diabetics. So to follow up and figure out what is going on, what is wrong, we went back, and as I mentioned before, the way the bacteria can go into the cell is either via coating with antibodies or coating with complement. So to quickly distinguish these two possibilities, we went ahead and heated the serum. So we heat and activated the serum, and that knocks out the complement. That way, we would only be evaluating entry via the antibodies. And the question is, after we do that, do we still see a significant difference between the diabetics and the non-diabetics? So this is a table with the summary. This is the classification of the participants, those without diabetes. When we use the fresh serum, which means entry via antibodies and complement, we observed about roughly 1/3 of the monocytes go in. This is what we had observed, which is significantly higher than people with diabetes. That's the p-value. When we heat the serum, so we only have entry via the antibody mechanism, there's a dramatic reduction in the percent of phagocytosis, which is to be expected because we're only seeing entry via the antibodies, but that difference is no longer significant. What that suggests is that the problem in diabetes seems to be the entry via the complement pathway. So what we did next was to determine is the defect, the defect could be in the monocyte complement receptors or in the serum complement. So we began studying the entry, the defect in the monocyte itself. So for this, we had to stay away from the mycobacteria because the mycobacteria can enter via the complement or the antibody. So we wanted to distinguish the two possibilities. So what we did is instead of mycobacteria, we took sheep red blood cells. If we incubate sheep red blood cells with monocytes, nothing happens. If we coat sheep red blood cells with complement and add the monocytes, they will be phagocytosed, and these red blood cells will be phagocytosed via the complement receptor. If we coat the red blood cells with IgG, they will enter the monocytes via the Fc receptor. So this would allow us to understand the mechanisms. The other thing is that when we're doing these experiments, we don't need serum from the diabetes patients. We're strictly evaluating the monocyte function. This is an example of what those experiments look like. And so the blue is all the monocyte nuclei, and the red are the red blood cells stained with a red dye. So in this case, we have 10 monocytes, out of which 8 have phagocytosed at least one red blood cell. So this is an example of 80% phagocytosis. So the question is there a difference in the monocyte function between monocytes from diabetics and monocytes from non-diabetics? And these are the results. So I stratified them here. I showed them by HbA1c, and then the diabetics are those with the gray dots. The non-diabetics are the black dots. And what you can notice is that in diabetes, and particularly with a higher HbA1c, there is a reduction in the capacity of the monocytes to phagocytose the red blood cells. We thought the defect would be only in the complement pathway, but to our surprise, we also find that there's some level of defect in the Fc pathway as well. So in conclusion, the monocytes themselves have defects in the major pathways for phagocytosis, via complement or via Fc receptors. So one possibility is that the monocytes have lower expression of the surface receptors to get the red blood cells in, or they have a functional defect. They may have the receptors there, but they don't work. So we evaluated the phenotype. Is there a difference in the expression of the receptors? And I have here the differences in the density of expression of 2 of those receptors, but the findings were there is no difference in what the monocytes express in terms of the receptors. We also evaluated, is there an increased expression of receptors that inhibit phagocytosis? And we thought this receptor here could be higher in the diabetics. And the opposite was observed. It's even lower. So the monocyte is saying, I'm ready for phagocytosis. I'm not inhibiting phagocytosis at all. Despite that, the phagocytosis in the diabetic monocyte is compromised. So in conclusion, there seems to be a functional defect in the monocytes from the diabetes patients. What we are doing next, and I'm just going to, so one of the things is we're finding a defect in the monocytes. We're also exploring if there's a defect in the serum components, in the complement or the antibodies. Remember, diabetes is a systemic disease, so it may not be affecting one aspect of the immune response. It may be affecting multiple aspects. So we're looking into that as well. And then finally, if we're seeing this defect in phagocytosis, what does that mean in terms of the survival of the mycobacteria? So phagocytosis is an incredibly complex process. It requires a lot of energy for the cell to engulf the bugs. It also, and then it is coupled to the killing of the microbes, generally speaking. So there's oxidative stress induced. That was the original goal of phagocytosis. So the fact that the mycobacteria is entering in a different way to the phagocyte may mean that whatever happens inside the cell is different in the diabetic monocyte versus the non-diabetic monocyte. So we went ahead and we have begun evaluating what's the fate of the mycobacteria inside a diabetic monocyte. Is it going to have a survival advantage, or is it going to be contained or killed just like any other monocyte? And this is very preliminary data. As you can see, there's very few participants. But what we've noticed is that this is the mycobacterial burden inside the monocyte 24 hours later. And it suggests that perhaps in the diabetic monocyte, there is better opportunity for survival of the mycobacteria compared to the normal monocyte. So we think that there seems to be an advantage. So putting everything together that I've told you today, plus what is known from some mouse models, what we observe in diabetes is what appears to be an underperforming innate response. That's the first encounter with the bug. The innate response is not working well. The result of that may be that it allows the bug to replicate more. There's higher bacterial burden. As a consequence of that, you're getting a hyperreactive cell-mediated immunity. And we saw that reflected in more interferon gamma, more IL-2 secretion. Whether that response is efficient or not, we really don't know. And so if it's inefficient, it may even promote or facilitate more bacterial burden. Clinically, the consequence of this is that a diabetes patient may be more likely to have latent TB infection. And the good news is that because there's good production of interferon gamma in the clinical setting, we can identify the diabetes patients at this stage. Then it is very likely that all of this combined contributes to the higher risk of tuberculosis. But as you can see, this is a very simple cartoon. We still have lots that we don't know before we can take these basic science observations and have them make a bigger impact on how we manage the tuberculosis, how we prevent tuberculosis in the diabetes patients. So with that, I want to thank my team. Diana Gomez did most of the studies that I presented. Marcel Twahirwa is the local endocrinologist in South Texas. He identifies the diabetes patients for me. And Larry Schlesinger is my collaborator who has shown me a lot of the monocyte biology. Thank you.
Thank you, Blanca, for making that very complex things look so simple. Now let me call on Dr.